Unit 6, Energy and Biology. Isn't energy for physics? Organisms use energy to carry out many tasks of living. So things like growing, reproduction, movement, transporting things in and out of the cells all require energy. So what is energy? Energy is the ability to cause change. There are many different forms of energy, from mechanical, light, sound, thermal, electric, and so forth. Even though energy comes in many different types, there are two basic forms, which we kind of think of as kinetic or potential. Kinetic is energy an object has because it's moving. Potential is energy an object has stored because it's position or condition. So for example, if something is higher up, it might have a higher gravitational potential energy. If I pull a rubber band back, it has a higher elastic potential energy. And just some foods have different chemical potential energies. You get more energy from eating different types of food. So within potential, there could be many different types. And then also, don't forget about these laws of thermodynamics that you learned last year in integrated science. First law of thermodynamics, you can't create or destroy energy, it can only change form. So anytime energy changes form, some of it is turned into thermal energy, which we refer to sometimes as heat. Most organisms, this is not very useful. You can't really recapture that heat and turn it into another type of energy. It usually is kind of dissipates or lost through, to the universe when this occurs. The second law of thermodynamics, also known as entropy, all systems tend to greater disorder, or entropy, unless energy is added to the system. So I kind of relate this to your bedroom. If I just keep throwing my clothes after I wear them onto my floor and so forth, my room is going to get messier and messier and messier unless I add energy to the system and clean up. Usually greater entropy means greater stability, which means that, if you think of it like energy that's being released into the system, if I think of like an atomic bomb, there's a lot of energy at that one main spot, but then over time as it's released further and further through the universe to out of your immediate point, now we have a greater stability. These laws apply to all systems, including biological ones. So how does this energy apply to biology? All living things need a constant supply of energy to fuel the activities of life. All energy for living systems comes directly or indirectly from the sun. We've talked about this in our previous notes, so I'll just kind of read through this. Remember, organisms can be autotrophs, which harvest their own energy directly from the sun, or inorganic chemicals, such as chemosynthesis, or heterotrophs, which consume food, other organisms, to get their energy. Section 2, Energy and Chemical Reactions. Basics of chemical reactions. So a chemical reaction is when chemical bonds between atoms are broken or formed, therefore creating one or more new substances. We have two sides of a chemical reaction. We have the reactants. These are the substances that you start with or combine or break apart. So it's kind of like the ingredients. And we also have the products. These are the substances at the end, the new substance that is formed or produced. Most chemical reactions are reversible, not all. So here's a graph of activation energy for a chemical reaction. Chemical reactions need a kick to break existing chemical bonds, and this energy is called the activation energy. And I'll show that right here. This is the activation energy. So it's kind of like a kickstart. Think of it like your car if you have the ignition, the spark that starts everything. That's the activation energy. It's a kickstart to break existing chemical bonds and start the reaction. If you can lower this activation energy, so if I lower it right, show the arrow a little smaller, I've lowered the activation energy, and that just means that the reaction can happen at a faster rate, which can be good for an organism. Energy required for chemical reactions. So we have something called free energy. This is energy from chemical reactions that drive cellular activities. Reactions that release free energy are called exergonic. These products have less energy than the reactants. Cellular respiration is an example of this. Reactions that absorb free energy are called endergonic. These products have more energy than reactants, and photosynthesis is an example. One way to remember the two is to look at the front end of the word. Exer kind of looks like the word exit, which exit, you release something, it goes out. Ender looks like the word enter, which means it absorbs, it comes into the reaction. So that's one way that I remember those two words. Here is a diagram of an endergonic versus exergonic reaction. So if we look at a couple things here. Here we have the reactants. This is an exergonic. Remember, exer, exit, energy is released out. Okay, so if we look at this diagram right here, the reactants, and we compare the reactants to the products. On the y-axis is the energy released. And we'll notice that the energy starts here, and then the energy it ends there, which means we've kind of lost energy. We have a lower energy in the products than we did in the reactants. The reactants have a higher energy. Because the reactants have a higher energy than the products, energy is released, meaning it is exergonic. If I look at the second example, endergonic, remember ender meaning enter, or energy is invested or entered in. So if I look at this reactants are right here, this has a lower energy. And if I look at my products, this has a higher energy. Well, how did it get a higher energy? It had to have some energy absorbed into the reaction. Therefore, energy must be supplied. We have a positive number here. That means energy has entered the reaction. Sometimes you'll see this G as an H, and we think of that as heat. So the examples we do in class, we will refer to that G as an H for heat. All right, chemical reactions and energy. Some chemical reactions release energy. Again, we said they are exergonic. Sorry. And one example is digesting polymers. So digesting molecules equals less organization or a lower energy state. Some chemical reactions require energy or an input of energy. That means energy is entering, endergonic, such as building polymers. Building molecules would mean more organization or a higher energy state. So what are catalysts? Catalyst is a substance that changes the chemical reaction so it can proceed at a lower activation energy. Remember, when we talked about that activation energy, if I can lower it, that means the reaction can occur faster. These substances are not used up or changed by the reaction. They're just kind of like something that helps start the reaction. They do not make the reaction spontaneous, meaning the reaction can't just occur. So here's an example of our graphs again. You look at, this is the same reaction. In the blue, it shows, here's the reactants, and it shows the amount of energy that's required when you do not have a catalyst, without a catalyst. Then in red, it shows the same reaction, but you'll notice it required a lot less energy, and that's because it has a catalyst. So catalysts will lower the activation energy. This is the activation energy. And this is the activation energy for the blue. So you can see that the red has a much lower activation energy because of the catalyst. So catalysts reduce the amount of energy to start a reaction. And again, this is just showing the uncatalyzed versus the catalyzed reaction. And the new activation energy is much less. Phew, it takes a lot less energy when you have a catalyst, which means the reaction can occur a lot faster. So what catalyzes biological reactions? Most living things cannot tolerate the amount of heat needed to make a reaction as quickly as they need to. Meaning, I would have to have so much heat entered into a reaction in order to get the reaction to occur that the organism couldn't tolerate and could possibly die trying to have that occur. This means they need a catalyst because they need to lower the amount of energy, so they don't need as much heat for that reaction to occur. Enzymes are catalysts that are used by cells to trigger and control biochemical reactions. So an example of an enzyme is called amylase. That's a catalyst that breaks down starch. One thing to note about enzymes is they usually end in ASE, and that's the name. Enzymes as catalysts. So they have a substrate, which is the reactant to which an enzyme binds to. Enzymes can only bind to certain substrates. The place where the substrate binds is called the active site. Substrates that don't fit won't bind. Without enzymes, chemical reactions necessary for life would not occur at a rate sufficient for sustaining life. Meaning, if I didn't have the catalyst enzyme, the heat needed would be way too high for that organism to sustain itself. And that reaction wouldn't occur. Therefore, the organism would not live. So again, we have the substrate, which is the purple part right here. This is the substrate. And right on where that connects, that puzzle piece is called the active site. Oh, I'm sorry. I made one mistake. Sorry. The substrate is the yellow piece. Apology. The enzyme is the purple. The active site, the substrate, binds to the enzyme, and it forms two new products in this case. So again, the enzyme is the purple, the substrate is the piece that binds to the enzyme, and it binds at the active site, and it produces two new products. 3 properties of enzymes. They have a lock and key, which means they are reaction specific. They are not consumed. A single enzyme can catalyze thousands of reactions per second. And they can be denatured, which we'll talk about later, but denatured means it can change its shape. So shape changes, which means they won't work. What causes the shape to change, we'll talk about later as well. So lock and key model, meaning a specific substrate has to fit in a specific enzyme. If they're not the right shape or they're not the right substrate, they will not fit. Influencing enzyme effectiveness. So temperature. Each enzyme has an optimal working temperature and range. If it's not in that range, the enzyme changes shape, which we call denatures. When it denatures, it's no longer, because it changed its shape, it's no longer effective, and therefore it will not work. Enzymes also work at an optimal pH. Each enzyme has this optimal working pH range. If it's not in the range, the enzyme, again, changes shape, which means it also denatures. Therefore, it doesn't work.